Hey everybody, we're going to talk a little bit about the cell in this particular video. And I wanted to just draw your attention to that this here is the cell in all of its glory. And if you look up here, this is your mitochondria, right? So one thing you want to understand too, that this is you have a part side one, side two. This side has just been cut so that you can kind of see, and this side has not been cut. So there's really no difference between this side and that side. But with all that being said, this is the mitochondria. You can see that one that's going to be on this side as well. And this one is going to be involved in making energy. So it's going to take all the glucose from the food that you consume and make ATP. This structure in yellow, not in yellow, but in blue, that sort of surrounds this. This is going to be the cytoplasm or the cytosol, depending on what you're referring to. So in my particular class, if I'm pointing to this, you can say cytosol or cytoplasm. I am not going to. You don't have to differentiate them. But if you are supposed to differentiate them, just know that the cytosol is the actual fluid, right? There's a fluid within the cell, and the cytoplasm is really this whole space, right? So if that sounds confusing, kind of think of it as a swimming pool, where the swimming pool without the water is the cytoplasm, while the fluid inside of there would be the cytosol. And so when you have the cytosol and we have the fluid inside of the space, all of that will be referred to as the cytoplasm. So it sounds a little bit confusing. It's in your book. You don't have to be able to differentiate it for my class, but if you're in any other class, you may have to. Anyway, if you see these invaginations that come up on top of the cell, these are going to be called villi, and they're going to assist in the absorption of nutrients. This structure here is going to be the nucleus, the purple, and the little holes are going to be called nuclear pores. And the structure, the ball within the ball, this is going to be the nucleolus. So the nucleolus, the nucleus, the nuclear envelope, and I'm kind of zooming in and see these little squigglies, right? That's actually DNA or chromatin, right? So that's chromatin. Okay, so surrounding the nucleus, you're going to have this structure called an ER. And the ER comes in two flavors, chocolate and vanilla. Just kidding, just kidding. It comes in rough and smooth. So this is going to be rough, and I can actually touch it here, and it feels kind of rough. And the reason it's rough is because it has ribosomes docked on them. And on this part will be smooth, and you can tell that it's smooth because it doesn't have any ribosomes. So rough ER, smooth ER, and this up here would be your Golgi. You can see that up here. So nucleus, rough ER, smooth ER, and Golgi apparatus right there. You have lysosomes or peroxisomes. You do not need to be able to identify them. What I would tell you is that peroxisomes are going to be involved in detoxifying the cell or the blood, like they found in the kidneys, for example. And lysosomes are going to be involved in breaking down worn out organelles. So, for example, you don't have the same mitochondria that you did when you were a baby, right? There's a lysosome that broke that down and then made you a whole new mitochondria, right? The last thing that's on here are going to be, these are going to be called centrioles. They kind of look like little yellow licorices, kind of. And they're going to be involved in moving or the moving of genetic material during mitosis or meiosis, which we will talk about in just a second. So, so I think this is it. If this is talking about tissues, this cell actually sits on what's called the basal lamina. So you can see that's there. And then this is kind of showing you this will be like another cell that it's attached to. Okay. The only other thing that I would tell you that's actually not on here would be the cytoskeleton. So the cytoskeleton would be surrounded around here everywhere, but it's not on here. So you do not have to differentiate for the practical or the quizzes the components of the cytoskeleton. So you don't need to know about intermediate filaments or microtubules or microfilaments. You just need to be able to identify that, hey, these are going to be a cytoskeleton. Okay, so hopefully that was helpful.